All right. All right. Thank you, everyone, for coming today. I'm really excited to talk about the work that I've done over the last 5 years here at University of South Carolina in the lab of Dr. Michael Gower in the Department of Chemical Engineering. So without further ado, the title of my talk today is Polymer Microparticles for Encapsulation and Presentation of Anti-Inflammatory Agents. So to begin with, I'd like to talk about some broad background for our work, and it really has to do with inflammation and inflammatory diseases. So as we can see in this graph on the right, numerous diseases promote chronic inflammation, and that chronic inflammation can in return really exacerbate those diseases that it underlies. And this includes some notable diseases that we're all familiar with, such as cancer, cardiovascular disease, autoimmune disease, muscle atrophy, and obesity. And so really strategies that address that underlying inflammation in these diseases can have a lot of promise. And strategies that include more established techniques like administering anti-inflammatory drugs, and then also reprogramming the immune system to become more anti-inflammatory in response to that disease. And so two very prevalent and related inflammatory diseases that I'd like to highlight are obesity and muscle atrophy. And the reason I say that they're related is that many times with obese patients, they become more sedentary and they suffer from muscle atrophy. And patients with muscle atrophy who tend to be elderly are also sedentary, and that promotes obesity. So they often go hand in hand. And so to get into some statistics here, obesity, which is defined as having a BMI over 30, is characterized by an inflamed adipose tissue. And it is a comorbidity for heart disease, cancer, diabetes, and stroke, which are all leading causes of death in this country. And as we can see on this graph on the right, the prevalence of obesity has really continued to rise over the last 20 years, from 20% in the year 2000 up to 42% in the year 2018. So it's starting to approach half the population being obese. And as of 2018, 9% of the population was considered severely obese, which is having a BMI of over 40. So the prevalence continues to rise, but few treatments exist beyond diet, exercise, and appetite suppressants. And these are prescription medication. And in some extreme cases, bariatric surgery, which is very invasive. And so despite these treatments, the cases continue to rise, so new strategies are needed. In addition, muscle atrophy is also on the rise, along with the rise of obesity, but also as the median age in the country continues to increase. It keeps setting records. The number of people in retirement age are also keeps increasing. And so in comparison to obesity, few treatments exist beyond exercise and supplements for muscle atrophy. And this includes protein and vitamin D, which have been around for a long time. But really my point here is that anti-inflammatory drugs may really benefit these diseases, attacking that disease at its core root, inflammation. But it can also have limitations, which I'll get into. So the drug that we've been interested in is resveratrol. And the reason is that it's this anti-inflammatory polyphenol found in grapes and peanuts, and it's thought to be the beneficial property behind red wine. But it's also been shown to have anti-obesity effects in cell culture. So given to adipocytes, it causes lipolysis, which is the breakdown of lipids. And so a lot of promising results here. And also it was reported that resveratrol alleviates obesity-induced skeletal muscle inflammation. So both in the dish and in small animal models, it was shown to be a very effective treatment for obesity and muscle atrophy. But the problem is that when it's given to humans, it's pretty lousy. So in clinical trials, it's been shown to have low bioavailability, meaning it's just not getting to the tissue of interest at high concentrations. And scientists have, addressed this problem in the pharmaceutical industry for some time, and one solution is drug delivery devices. So our objective was to design microparticles to control the release of resveratrol for this very purpose. So getting into polymer microparticles, the perspective is that they were kind of conceived in the 1970s, and many polymers were tried during this time. And one of the more promising ones to emerge from this time period was this one right here, polylactide coglycolide, or PLG. which is a copolymer of lactic acid and glycolic acid. And the reason that it's so effective is that it is biodegradable, it's biocompatible, and it has tunable degradation, which is controls that release rate. And by 1989, the first polymer microparticles were FDA approved. And so this figure here kind of demonstrates the real benefit of a control release system. That with an oral tablet, you get this spike in drug concentration that then diminishes, requires a second dose, you get more this sinusoidal effect. Whereas with a controlled release system, one administration provides control release over a period of time. So that's really what we're shooting for. And this list is a list of FDA-approved PLG-based drug delivery systems. And they've been FDA-approved, about 20 of them have been FDA-approved for a variety of diseases, from prostate cancer to breast cancer to schizophrenia to opioid dependence. And what this review article that has tabulated this, this table mentions is that the drug delivery field has really exploded over the last few decades, but not that many formulations have made it to FDA approval. And in fact, the fabrication technique for these formulations is limited as well. And the most commonly used one is the oil and water emulsion solvent extraction method for producing these particles. And one reason the author mentions is that this method has withstood a lot of the rigorous demands of FDA approval, showing reproducibility and scalability of your system. So that's the system we chose to use, and I'll go through it right now with you because it's what we do in this talk. We have drug and polymer dissolved in organic solvent, which is then mixed with water with polyvinyl alcohol, which is our emulsifier. At high speed, it forms a primary emulsion. And that primary emulsion can be brought to a larger volume of water, which promotes the extraction of the organic solvent. And that extraction of the organic solvent causes the precipitation of the polymer and the drug into microparticles. These microparticles can be centrifuged and freeze-dried, and what we obtain is this dry powder. And this dry powder, we can then take a look at it under scanning electron microscopy, and this is what we see. We can make particles on the size range of 10 microns that are smooth, spherical, and monodisperse. And we chose to use the same materials that are often used in the FDA-approved systems, which are PLG as our polymer, our emulsifier polyvinyl alcohol, which is a polymer of OH, and our solvent dichloromethane. And we use dichloromethane because it is immiscible with water and also highly volatile. So this brings me to my first aim, which was to develop microparticles for resveratrol delivery to the adipose tissue. And we were interested in doing this because although many formulations had been done before, we hadn't seen people trying to deliver resveratrol to the adipose tissue. So we thought this was the gap that we could fill. So to begin with, we went about designing our particles, and we needed to do solubility studies. And we saw that resveratrol was not soluble in DCM. So that's a problem. You need your drug to be soluble in your primary solvent. And what we found, without going through this too much, is that a ratio of 1 part ethanol to 3 parts DCM, using ethanol as a co-solvent, produced spherical PLG particles with high yield. And so going to panel D here, we were able to reproducibly make particles with a consistent size range. So we went ahead and made our particles with resveratrol. And to our surprise, we found that our resveratrol particles had this non-spherical morphology. And so comparing this to all the other published literature out there, they were all appearing spherical with drugs in them. So the question was, why is resveratrol causing this non-spherical morphology? We weren't quite sure. But we took resveratrol out of the system, and they produced smooth spherical particles. And taking ethanol out of the system also produced smooth spherical particles. And looking at the release or the loading of these, this resveratrol, it was loaded at a rate of about 65 microgram per milligram of resveratrol loaded, 6.5 weight percent. And then we did a release assay on these particles. And here's what we saw. We were able to get that sustained release that we want to see from a drug delivery system over 42 days. And looking at the first 14 days, we saw that we had sustained release over this time as well. Although there was some initial variable effects in these first couple of time points, which we thought may be due to the fact that we weren't getting, or the same morphology. You know, all these particles kind of look different under the microscope. So we wanted to show that the resveratrol loaded in these particles was bioactive. And what we did here is we stained our adipocytes that we grew up in a dish with lipophilic dye. And that lipophilic dye, we could then extract and quantify the amount that was remaining in these cells. And so resveratrol is shown to reduce this stain. So without going through this too much, panel H here shows that PLG particles given to these cells had a certain oil red O stain, and the resveratrol loaded in particles reduced that stain to a similar extent as resveratrol. And so this showed to us that our drug was bioactive. But we were really interested in that non-spherical morphology that we were seeing before. And so what we did here is we reduced the amount of resveratrol dissolved in the oil phase from 10 mg per mil down to 4 mg per mil. And what we saw is that at about a threshold of about 5 mg per mil to 4 mg per mil, we started getting smooth spherical particles again. So there was something about this threshold that was important to the morphology. And we also looked at the PVA percentage in the emulsion and in the evaporation bath. And what we saw was that as we decreased PVA, we regained that non-spherical morphology. So the emulsifier, which is not surprising, is the stabilizer in the solution, really is important to this morphology effect as well. So we took those spherical particles and we showed that we could control their size as well. So increasing homogenization time and speed decreased particle size. From 30 seconds to 5 minutes, and from 5,000 RPM to 11,000 RPM produced smaller particles. But what was more interesting about this was that we saw this correlation between loading and particle diameter. So as the size got larger, the loading of the particles got smaller. And we hadn't seen this effect before in the literature either. And so what we did is we did a release assay over 14 days, and we showed that the small particles released their drug over the first, had a large burst release over the first day, the first 6 hours in the first day. About 60% was released. Then there was a plateau over a period of time of 14 days. And the large particles had a much more slow, sustained release over that same time period. And so to kind of confirm that the resveratrol, so what is this suggesting? What's the difference between a small and a large particle? Well, the real difference is the surface area to volume ratio. That as these particles get smaller, they have more surface area per volume. And then we tried to confirm that this, in fact, was happening by washing our particles with ethanol. For 1 minute, we washed these particles, and we saw that with large particles, about 20% of the resveratrol was removed. And with small particles, nearly 80% of the resveratrol was removed. So in our minds, this had confirmed that resveratrol was loading on the surface, as we had predicted. And so to summarize my first aim, we showed that resveratrol is bioactive. Resveratrol causes non-spherical morphology, and we had control over morphology through resveratrol concentration and PVA concentration. And we had control over size, and we could produce different release profiles depending on the particle that we produced. So this resulted in my first authored publication, my first publication in International Journal of Pharmaceutics. And so this leads me to my second aim, which was to understand and control non-spherical morphology in drug-loaded particles. You might be asking, well, why are you so interested in these non-spherical particles? Well, the reason is that first of all, we weren't seeing other groups reporting this effect. You know, we had loaded other drugs in our particles. They turned out smooth and spherical. Other groups have done it. They're all smooth and spherical. So why is resveratrol causing this effect? And the second reason that we were interested in this is that morphology can really cause bioactive effects. You know, big names in the field have mentioned that in drug delivery, shape matters, and that wrinkling non-spherical particles was shown that they promote cell attachment more than smooth spherical particles do. And so, so really what I'm trying to say is that causing this morphology change in your particles can really promote cellular attachment. And going back to that list of FDA-approved particles, we think that resveratrol could be used as an additive in these emulsion systems to give your particles some texture that could promote cellular attachment. So that was really the motivation behind that work, why we're interested in this work. And so we took our emulsions and we looked at them under the microscope. What we saw is that, first of all, in the first row, that DCM particles shrank as the solvent was removed during the extraction step, and this produced smooth spherical particles. And emulsions with ethanol in them did the same thing, and so this has been reported before that this happens, and these particles form. However, with resveratrol, we take these smooth spherical droplets, and they started to crumple in on themselves, and they produced our little raisin particles that I showed before. And we could take these images and stitch them together under microscopy and show that here these droplets just shrink slowly as they were expected to. But when we looked at resveratrol droplets, they just kind of crumpled in on themselves. And so this was really surprising. And we kind of made an analogy here to a typical emulsion retains its shape because that surface is fluid. And with the resveratrol ones, we proposed that there was this hard shell forming on the surface of these particles that was then, as the solvent was extracted, that surface was deflating. So the question is, well, why is there this hard shell on the surface? Well, thinking about this droplet is there's a soup of spaghetti-like polymer molecules and drug molecules in this system. And so you could imagine that if we had organization of those molecules, there may be some crystallization that was happening at the surface. And so that led us to do some XRD measurements. And what we wanted to do here is kind of identify the crystalline nature of the material. And so that's what XRD can tell us. If you see sharp peaks in the spectra, you expect there to be crystals. So starting at the bottom, we looked at resveratrol. There was the sharp crystalline nature of the spectra, of the pattern. And so looking at PLG, blank particles, which are amorphous in nature, we see this broad amorphous peak. Going up to the top, we see PLG and resveratrol powder mixed together. We get this combination of the amorphous effect and crystalline effect. And then looking at PLG plus resveratrol, we noticed that majority of resveratrol was, as we expected to be, amorphously, was embedded in that matrix amorphously. But we did see these small peaks here, which could be indicative of that shell formation that we were proposing. Okay, so moving along, we also found that the amount of co-solvent can impact morphology. So going back to that first formulation, it had 25% ethanol and 10 mg per mil resveratrol. Looking now at 4 mg per mil, we saw this non-spherical morphology reemerge. So this was kind of surprising, and the reason is that at 10% ethanol, with the same amount of resveratrol, we formed these smooth spherical particles. So why was it that just by increasing the ethanol amount, we could produce this non-spherical morphology? Well, the reason could be that the ethanol is playing a role here in the extraction process. And so we investigated co-solvent a little bit further. We looked at ethanol, methanol, and acetone particles. And we saw that ethanol and methanol particles had very similar morphology. They had this raisin-like structure under the microscope. And then acetone looked very different. It had these jagged, indented structures that looked much more like sort of deflated ping pong balls. So we kind of wondered, well, do they have different surface areas on the surface of these particles? So we could do the BET surface area measurements. And here we have 1.2 meters squared per gram for the ethanol particles, 0.9 meters squared per gram for the methanol particles, and 0.7 meters squared per gram for the acetone particles. So it did seem that the surface area was reducing in response to the acetone, like we were suggesting. And then so the other question was, is co-solvent even important in this system? Could we switch to ethyl acetate, which turns out could dissolve resveratrol and be used as the primary solvent? And when we made particles this way, they were smooth and spherical, and they had a large loading of 50 microgram per mg. So we could also produce these particles. And then this led us to a question, which was, is resveratrol unique to this morphology? And that was the question. And so going through this, we took this molecule transtobine, which is very similar to resveratrol except without any OHs. And it has this spherical morphology with high mass yield. And then penicillin, which is resveratrol without one OH, it also had spherical morphology with a high mass yield. And then looking at resveratrol, which we've already looked at, the mass yield decreased a little bit. And then going to picoadenol, which is resveratrol with just one more OH group, the mass yield crashed. So the emulsion hardly formed in this case. And what we noticed with these molecules is that there was this correlation with log P value. So the log P is a measure of hydrophobicity. And as we got to more hydrophilic compounds, we noticed a sort of crash in the emulsion process. And it's something about the log P of 3 was causing this effect. But at any rate, with this study, to answer the question, was resveratrol unique for this process? The answer was yes. Only resveratrol produced the raisin particles with a consistent mass yield, and we could produce these particles consistently. So that was the answer to that question. And this kind of led us to a hypothesis of what was happening. So we turned to the literature, the physics literature, and we noticed that with that the morphology, the shape of a deflated shell was dependent on the extraction rate and the amount of volume that was extracted. And so these were some simulations done. And what we noticed is that these particles that they were showing in the physics literature were similar to the particles that we were making. So these look like our ethanol particles, and these look like our acetone particles. And so this would suggest that there was an effect of extraction. And indeed, if you look at acetone versus ethanol, the molar solubility of acetone is less than that of ethanol, suggesting that it should be extracting out of the system slower. So this leads me to the summary of aim 2, which was that we could produce spherical dichloromethane particles. We could produce highly crumpled DCM plus ethanol plus resveratrol particles, these indented DCM plus acetone plus resveratrol particles, and spherical ethyl acetate particles with high amounts of resveratrol loading. And this morphology change was dependent on co-solvent, solvent, and was unique to resveratrol, and we proposed this shell crumpling mechanism. And so this work was published last year in the International Journal of Pharmaceutics. But what we thought about this work was more broadly, resveratrol could be used as an additive in oil and water emulsion systems that are already FDA approved. And in fact, we filed a patent on this, which was approved as this new method for producing non-spherical particles. And so this leads me to aim 3, which was to develop modified PVA for PLG particle surface functionalization and macrophage targeting. So the goal here, kind of going back to the theme of inflammation, was to surface functionalize PLG particles in a new way to target macrophages, which are innate immune cells that are central players in the regulation of inflammation. And to do that, we wanted to look at mannose, which is this hexose monosaccharide, ligand of the mannose receptor on macrophages, and activation of mannose receptor is known to promote an anti-inflammatory phenotype. And so previous groups have actually attached mannose to particles. And this group showed that those particles co-localize with the mannose receptor. But their technique has not been implemented in those FDA-approved systems, which I always like to go back to. And so what we wanted to do was design particles with novel functionalization for attaching mannose and promoting macrophage targeting. So our idea for doing this was to modify PVA using isothiocyanate chemistry. And so you might remember that PVA in this emulsion is the emulsifier, which coats the surface of the particle. And it's been shown to do this. We can quantify it after freeze drying, that it's there in quantifiable amounts. So our idea was to attach ligands via PVA. And we could do this in a one-step reaction with isothiocyanate, which reacts with the alcohol in PVA and produces this monothiouretane bond. And so we chose to do this with 2 very colorful molecules to begin with, and then move on to mannose. And the colorful molecules are fluorescein isothiocyanate, shown here. And one nice aspect of this is that it's colorful, so we could see our products were gaining in color as we made these products, and also rhodamine isothiocyanate, which appears pink here. And to quantify this a little bit further, we did FTIR and NMR analysis. The FTIR showed that there was an increase in the C double bond C bond, which is indicative of FTSE conjugation onto this PVA. And then the NMR what we could see is certain peaks that were present in only PhD were present in our product and were not present in PVA. And we could actually integrate these peaks against the peaks in the backbone of the PVA, and we could calculate a molar conversion. And we got a molar conversion of 80%. So we were really happy and excited about that. So we went right ahead and we made our mannose particles. And this mannose PVA initially had, we did it the exact same way we did PhD, and it had a reaction conversion of 0%, 0.1%. So we were very disappointed about this and a little confused, but we did find that there was this temperature dependence of mannose with PVA. And as we got to 100 degrees C, we could approach 30% conversion, which wasn't 80%, but it was much better than what we were getting before. So we thought this was acceptable, and we moved on to make our particles. So as you remember, PVA, this was our big idea, was that PVA could coat the surface of these particles. And under confocal microscopy, which takes a cross-section of these particles, we saw that they were green rings on these particles. So this meant that PVA was present. We were really excited about this. And also under epifluorescence, the RITC particles fluoresced red. So that suggested that RITC was there as well. And we produced all of these particles and we imaged them under microscopy, and we got a size range from 2 to 4 microns with a coefficient of variation of less than 40%, which suggested that these particles were not too polydispersed, with a mass field of about 50%, which was suitable for our needs. And we could also load these particles with fluorescent dye, coumarin 6, which was very bright under the microscope and allowed for tracking of these particles in cell culture. So we wanted to show that our mannose particles bound to macrophages, and this is what previous groups had shown. So in a 4C cold binding study, which is where we bring our macrophages down to 4 degrees C, we could seed our cells and add our particles on day 1 and extract the dye 10, 20, and 30 minutes after we added our particles. And what we found was that we tried 3 different doses here, and we showed that the amount of MITC particles that bound compared to blank particles in the lowest dose was statistically significantly higher than the blank particles. Now, we didn't see as promising results in the next 2 doses, but we were at least impressed that we could get it at the low dose, enough to believe that our mannose was working the way we expected it to. So one reason for these higher doses that it might not have been working is that they simply, there might have been some non-specific binding of the blank particles on the macrophages. So this led us to a big assay that we wanted to do to prove that our mannose was having bioactive effects, similar to that resveratrol assay, although this one was a little more complicated. We took media here and then media conditioned with macrophages, and media conditioned with macrophages treated with particles. And 24 hours after treatment, we added them to muscle cells. And muscle cells are these multinucleated cells, and we could count the number of nuclei there to see if the muscle cells had grown. So this was our treatment regimen. We plated our cells, we added media on all these days, and the last day we fixed and we stained with myosin heavy chain. And this is what these cells ended up looking like. Our media-only group, the cells didn't grow very much. If we gave them macrophage media, the muscle cells grew quite a bit. And if we gave them macrophage media with blank PLG particles, they grew about to the same extent as the macrophages. But if we gave them our mannose particles, they grew even more. And so looking at this graph, we could count the number of myotubes with greater than 5 nuclei. And we saw that our macrophages and our macrophages plus blank were much higher than our media, but our MITC particles were the highest. So this was suggesting that our effects were working. Now, we didn't get statistical significance, so I acknowledge that these particles may not be as great as they could be. You know, we're having some effects, but they're not, you know, superb effects. But to that end, here's the last bit of data I'd like to show, is that we just ran a trial of MITC, and these were all 5% MITC loaded particles. This one we added 20% MITC, and it had a conversion of 40%, with in fact nearly an 8-fold increase in the amount of OH groups reacted when the MITC added. So this has a lot more MITC added. And what I'd like to mention is that we just made the particles with these, and we noticed that they aggregated, which means that there's some surface functionality that's been imparted on these particles that is causing them to aggregate. And so this is the summary of AIM 3, and we're almost done here. PVA can be modified We can use it, and this was our big idea, was that particles can be coated and we can see them under confocal microscopy. And we got functional effects in vitro, seeing that with the binding assay and with myotube assay. And so really one of our big ideas is that we can add many different R groups to that isothiocyanate to get any number of ligands onto the surface of particles. So this idea really, we have submitted another patent for this, which was approved for functionalizing the surface of particles. And this leads me to my publications. I have two first author publications that are in print, 4 co-author publications that are also in print out of this lab, and then I'm finishing up the last touches on this last paper, which we plan to submit to the journal Biomacromolecules, and of course 2 patents for the work that we've done. So with that, I'd love to acknowledge my lab, Dr. Michael Gower, who's been so influential, Candice, Kido, Nick, the students, PhD students in the lab are so great. Undergrads, Alex, Haley, Christine especially have helped me. Alumni, Michael Henley, Kendall Murphy, Maddie Spetz, our collaborations with Dr. Jabari, Dr. Safa, Greg, all the cores and facilities at the University of South Carolina, funding and resources at the VA, National Institutes of Health, and the University of South Carolina. And with that, I will take any questions. Thank you so much.